Good afternoon to all of you. I'm delighted to see all of you here. It really bespeaks the interest in this area, and the time is ripe for the Brigham to put a stake in the ground that this is an area that we're very, very serious about. So welcome to the Women's Brain Initiative Symposium. The Women's Brain Initiative really builds on the Brigham's longstanding focus on women's neurologic and psychiatric health. As many of you know, the Brigham is a leader in tackling the clinical and research challenges related to our female patients, with work underway in the Mary Horgan Connors Center for Women's Health, the Women's Neurology Program, the Division of Endocrinology, the Department of Psychiatry, among others. So the launch of the Women's Brain Initiative really represents a further milestone and a commitment to this critical and growing field. Like Dennis, I want to give thanks to the program's generous donors, Nancy and Rick Moskowitz, for their vision and their commitment to understanding why many brain diseases affect women differently than men and how female-specific factors affect brain health. As many of you know, I'm a cardiologist. I've had a long-standing interest in caring for women with heart disease, and when I was at the NHLBI, funding research programs to try to understand sex differences in heart disease. I also was a proud supporter of the Red Dress public awareness campaign to raise awareness for women about their risk for heart disease. And as I've long told Dennis, if I were growing up today and starting my career, I would probably be a neuroscientist rather than a cardiologist. This is the burgeoning area. And I really hope to see the Women's Brain Initiative really accomplish many of the milestones in helping women understand their risk for various types of brain diseases. And hopefully we can bring this to a public awareness stage as well. So I really look forward to continuing to work with you in this area. We're off to a great start. I'm super excited. I wish I could stay with you for the afternoon, but I have my Board of Trustees meeting that I have to head off to. But I'm going to look forward to a report, and please know that you have my full commitment to making this initiative terribly successful. So thank you again.
Thank you very much, Betsy and Dennis. We appreciate it. So just two quick housekeeping items before we start. In the program, the title for Art Arnold's talk should be The Role of Sex Chromosomes in Causing Sex Differences in the Brain. So please note that change. And also, Charles Jennings will be orchestrating the Q&A. We hope to have plenty of time for questions after each talk. And so we encourage you to ask questions. There'll be two mics, which we ask you to speak into because we are taping this and there's a simulcast going on for those who cannot fit into this room. So with that, I ask Peg to join us. Thank you, Peg.
Well, let me start by saying what a tremendous honor it is to be a part of such an exciting new initiative. I really think it's a first of its kind. I don't believe that there's any other in the country. You hear a lot about women's health initiatives, but not so much about women's brain initiatives, so it's very exciting. Okay, so since our emphasis here is on the women's brain and women's health, I thought, of course, I'm going to immediately switch the topic to men. Because, so I didn't get the memo. All right, so it's not easy being male. Well, why would you say such a thing, particularly in the current era? Well, that's because it's not easy being a male when you're young. So males are conceived at a much higher rate than females, but they die in utero at prodigious rates. They're more likely to be born prematurely. They're more likely to suffer an injury at birth, even if they go full to term. And if they do suffer an injury, and so does their sister, they're going to have a far worse outcome. Even more broadly, they're much more likely to be diagnosed with neuropsychiatric or neurological disorders with origins in development. So these include autism spectrum disorders, ADHD, dyslexia, stuttering, Tourette's, any kind of speech or language impairment, and early onset schizophrenia. So the big question is why? Is this because females are resistant, or is it because males are vulnerable, or is it some combination of the two? So this is why I focus on sex differences in the brain, is to try to get insight into these particular questions. So how are brain sex differences determined? There are three main sources. First is your genes, and we're going to hear about that from Art Arnold. I was very upset when he had hormones in his title, because that's my purview, not his. There's obviously also your environment, and then, of course, there's your hormones, and that's what rules my world, and I'm going to talk about mostly. However, in humans, we can't make it that simple. In humans, environment and experience are likely, it's arguable, but are likely dominant. But more importantly, they are impossible to control for. Every individual has a different experience in a unique environment. So if we want to get to the biological basis of sex differences, we need to eliminate those environmental and experiential effects. And so we need to turn to our animal models. And these are some of the most favored animal models over many years. I'll just point out that I studied the increasingly exotic laboratory rat. This is not a mouse, it is a rat. And it has been a favorite model for over 50 years, which is why I continue to use it. So how do hormones determine sex differences in the brain? Well, they start very, very early on. So we all start life with potential, with the bipotential gonads, just little schmutz of cells near the unogenital sinus, which I'm sure well known to everyone here. And that bipotential gonad is by default going to develop into an ovary. And by default, I mean there's no clear genetic program that we know of. There's no requisite gene. However, if there is a Y chromosome, which has an SRY gene on it, which we're going to hear more about later today, you'll divert away from the default program to the formation of a testis. And that testis is going to produce testosterone, and it can be aromatized to an even more potent steroid hormone, estradiol. Now, the brain can also be viewed as bipotential as it's developing, and it also, by default, is going to develop into a, quote, female brain. And I'm using this very loosely because there's no such thing as an overall female brain, but the process of brain feminization will occur in the absence of an ovary or a testis or an SRY gene. However, if you have those high levels of testosterone and estradiol early in development, this is beginning in utero, it's complete in utero in humans, in rodents it extends into the first couple days of life, you will wind up with a masculinized brain. It's Mother Nature's way of assuring that gonadal sex and brain sex are in sync. Mother Nature doesn't give a whit about your chromosomal sex. She just wants to make sure that whatever reproductive physiology you have is going to be in sync between the gonads and the brain. So we have some fundamental questions that we want to answer then, which are, what are these sex differences in the brain? This is something, to me, always seems sort of intuitively obvious, but reporters tend to always, what are the sex differences? I'm sort of like, well, they're kind of everything. How are they generated? That's the question about hormones versus genes versus what are the signaling molecules. And then how are they maintained from this very, very early developmental event, which is basically when the brain is sort of a gelatinous mass, and then we're going to actually impact adult physiology and behavior. So to answer these questions, I've been interrogating two different systems in some detail, rather than sort of using the brain as a unitary unit, kind of digging down into specific brain regions that we know a lot about, and we know a lot about the behaviors and the physiologies that they control. All right, so in part of interrogating these systems, with a preamble that we now know that just about any endpoint in the brain is going to be modified by sex in some way, somehow. So this is sort of my cartoonish view of everything that happens in the brain of any significance. You have conception with neurulation, you have neurogenesis, migration, dendritic growth, myelination, glial genesis, phenotypic differentiation, synaptogenesis, synaptic pruning, and apoptosis. That's pretty much all there is, right? Every single one of these endpoints at some part of the brain at some time is modified by sex, either by hormones or chromosomal sex. I'm going to focus on just two. One is the formation of synapses, rather fundamental to brain development, and the other is neurogenesis and glial genesis. So the first one is synaptogenesis, and we're going to focus on the preoptic area because it is the major brain region controlling male sexual behavior. If you lesion this brain region, males lose all interest in sex. If you stimulate this brain region, males lose all interest in anything but sex. So it's a real nice kind of a connection between a brain area and a behavioral output. And we discovered quite some years ago that if we looked at the dendritic morphology of the POA neurons in males versus females, we found that the males have twice the density of dendritic spine synapses, which are excitatory synapses, compared to the female. This is not what a preoptic area neuron looks like. I actually borrowed this from a publication. They're not even very colorful in the preoptic area, but they do have lots of dendritic spines. And we can correlate that spine density to the behavior. And this is just part of a circuit, just so you know it's not floating out there in the Netherlands. It's part of a circuit. They get olfactory input from the female. It gets processed through the olfactory system, goes to the medial amygdala, BNST, and preoptic area. And if you have sufficient dendritic spines, you'll show male sex behavior in presence of a female, and if you don't, you won't. And so we found that treating females with a masculinizing dose of steroid as neonatal pups will increase their dendritic spine density to that of males. So what's the mechanism? So you would, of course, if we're talking about synapses, it must be a neurotransmitter, right? It makes perfect logical sense. And we and others spent many years exploring whether or not that was possible. You can actually sort of trace the whole history of neurotransmission, of people trying to look for it as an explanation. All right, so skipping right ahead, you would think it would be a neurotransmitter, but you would be wrong, because it's not. It turns out it's something completely different and unexpected, that actually this whole process, the signaling process, is initiated by prostaglandins. Yes, it's those prostaglandins, the ones that give you a fever, make you feel achy and sick when you have the flu or a cold, and they are normally associated with inflammation. They're what you take your aspirin and your COX inhibitors for. And through a series of studies, MD PhD student in the lab, Stuart Amato, demonstrated that testosterone and its aromatized product estrogen would increase would increase COX-1 and COX-2 production, making more prostaglandins, which would then stimulate neighboring astrocytes, and that would then stimulate the formation of dendritic spines on neighboring dendrites. And along the way, a new graduate student, Christopher Wright, discovered that a single injection of PGE2 on the day of birth to a newborn female, we're talking a little pinky eraser there, was sufficient to masculinize her dendritic spine density and her sex behavior for life. Just a single injection would raise her to adulthood, and she acted like she was a male. And this seemed absolutely extraordinary to me. This seemed like Mother Nature playing with fire. That you're going to take something as fundamentally important as reproductive behavior, and you're going to reverse it for life by a single prostaglandin injection. So we said, we must be lighting a fuse, right? We must be beginning a reaction that is going to burn for a while in order to change the morphology of the brain and behavior. So we asked, is there a positive feed-forward mechanism as a result of this single PGE2 injection? And is there another cell type that might be making prostaglandins? And just like everybody else at this time, we had been completely ignoring microglia, and suddenly, aha, the scales fell from our eyes, and we discovered this cell that's very poorly named, in my opinion. They're not micro because they're one of the most numerous cells in the brain, and they are not glia. They are actually immune cells. Right? They should have been named something else. Unfortunately, they were named over 100 years ago, so we're stuck with it. They also both respond to and produce prostaglandins. They take on a whole bunch of different types of morphology, which are a great readout of their mood. When they're thin and ramified like this, they're what they're called surveying or resting, versus if they're in this more amoeboid shape, they're very activated. They're cranking out a lot of cytokines and prostaglandins. We have been really rethinking our vision of microglia. A lot of that work being done here at Harvard, and realizing that they're very active cells. They're not just sitting there quiescently. We no longer refer to them as quiescent. They're constantly serving, checking on the brain. Every microglia sort of has a stable of neurons that it's in charge of, that they check on all the time, and they say that it takes about 2 hours for your whole brain to be surveyed by your microglia. So we just asked a simple question, is there a sex difference in these microglia in the preoptic area? And this just gives you a visual answer to that question. You can see that these male microglia, they are not checking, surveying. They are just not paying attention. They are all just sort of blebby and amoeboid-like, and they're sitting there kind of in an activated sort of angry state, whereas the females are nice, filigree, lacy, very much checking on everybody around them, taking care of all of their progeny. So what are these? I know it's really, even the microglia are gender typical. So what are these cells doing to help build the synaptic profile difference? So now I'm going to summarize a whole lot of data into just one animation, as opposed to showing you endless numbers of histograms. And apologies to the graduate students whose entire thesis have been reduced to just a single portion of this. We have here a big yellow neuron, a blue astrocyte, a neighboring microglia, and a neighboring dendrite. So the first step is testosterone will be falling out of the sky, and it will be aromatized to estradiol inside the neuron by the aromatase enzyme, bind to the estrogen receptor, translate quick to the nucleus, where it will transcribe the COX-1 and 2 genes, which are going to convert arachidonic acid into PGE2. PGE2 is then going to activate EP2 and 4 receptors, which are cyclic AMP linked, which are going to phosphorylate PKA, which is going to act on the GluR2 subunit of the AMPA receptors, causing them to cluster at the membranes. Additional prostaglandins now from the microglia are going to activate the astrocytes and cause calcium-dependent glutamate release, which is going to bind to the AMPA receptors and cause the formation and stabilization of dendritic spines. Thank you. behaviorally. But wait, there's more, right? So that yellow circle is not supposed to appear until right now. There's another cell type lurking there in the distance. So right now we've just shown that an immune cell is a critical player in the building of the synapses here. Mostly you hear about microglia pruning away synapses. This is the case of microglia building synapses. But there's this other cell type, this fourth cell type, the mast cells lurking in the background. What are mast cells? They are the other innate immune cell of the brain, only they come from the bone marrow with a myeloid precursor. They migrate to all tissues in the body, but there's also a brain resident population that stays in the brain, and they are the allergic response in your periphery and your first responders in your brain whenever there is an injury or an insult. So we, for reasons I won't go into, became very interested in these cells and went and counted them in the preoptic area. And you can just stain them with toluidine blue, right? You don't have to do a lot of fancy stuff. And it kind of looks like dirt on your slide. This is the preoptic area right here. And here's how you count mast cells. 1, 2, 3, 4, 5, 6. Let's go to lunch. Everybody in my lab wants to count mast cells. This is the easiest thing to do. We literally count them in the tens. But when we count them, as few as they are, we consistently find across development that the males have more than the females, quite a bit more than the females. And this is all during that early critical period for sex differentiation of the brain. Then they kind of go away and stay at very, very low levels. This is mast cells in the neural pill, not in the surrounding meninges. So we asked the question, are these mast cells talking to the microglia, which is what they do in the injury model? And the answer is obviously yes, that they are. And I'll just show you here that this is that same sex difference you saw up on the top. I'm going to try to use this. Up here on the top, there's the male, there's the female. Here's a male who we just told his mast cells to calm down and stop degranulating and stop releasing histamine for 2 days. And here's a female who we activated her mast cells with a degranulating agent, and they dumped out histamine. We know histamine is a signaling molecule, and you can see they're indistinguishable from the males. Their microglia are all activated and worked up. And what they're doing is they're releasing prostaglandins, and then that's driving this whole differentiation process. And I won't show the whole data, but we can get all the way to masculinizing the behavior of these animals just by manipulating their mast cells. So there are 4 cell types involved in the masculinization of this brain region, and 2 of them are non-neuronal origin, and the least plentiful, this mast cell, is the boss. This is the driver of the whole process. Okay, so that's one kind of example of where we can get unexpected origins of sex differences in the brain. But now switching to another system is the medial amygdala. And here we wanted to look at a more social behavior that does not have the baggage of involving sex, right? Because it turns out the NIH does not really like to give you money to study sex, but they're happy with play. And we sort of rediscovered play behavior. Play behavior used to be a very favored social behavior endpoint back in the day, and I'd say this is ancient history because this was published in 1986 in Brain Research. Remember that journal? And this is actually, I don't know if you can read this in the back, but yes, that is the Michael Meany and the Bruce McEwen who published this paper long ago. And they showed that the medial amygdala is the brain region that regulates the sex difference in play. So I'll just talk about that again in a second. But this play behavior is wonderful because it's very complex, right? It involves pouncing and chasing and pinning and solicitations and boxing and all sorts of it's a very rich behavior to study. That's one of the reasons why we like to study it. It occurs during a very restricted developmental window prior to puberty. Once they hit puberty, they lose all interest in play. Then it's all about sex, basically. Sex, aggression, territoriality in these animal models. It is more physically intense and frequent in males. This sex difference is established early while expression of independent of hormones. So it's organized developmentally in utero, but then they play as juveniles. In a rat, this is about a 25 to 30-day-old rat. And then it has strong face validity with humans and other species, because in humans we have both males and females play, but males play more physically and more intensely than females do, or boys versus girls. All right, so we're going to study on the medial amygdala. We're going to focus on the medial amygdala. The other thing we like about this play behavior is it's stable over time. So we measure them for like 10 days straight, and you can see that consistently males will play more than females. Females do play, they just don't play as much. And interestingly, we know that sex difference is controlled at the medial amygdala. So here we looked at another endpoint that we just lost. So our endpoint here was neurogenesis and gliogenesis as opposed to synaptogenesis. We didn't find any big sex differences in synaptogenesis. So we had found about 10 years ago that actually there were more newborn cells in the medial amygdala of females than males when we looked at 4-day-old animals, just birth dating with BRDU. And we were able to correlate this with the juvenile play behavior. And we also knew that it was mediated by, so then we found that if we treated females with a masculizing dose of steroids, it would decrease proliferation to that of males. Everything we do is hormone people, is we always first treat females with male hormones. And you would again think that the mechanism must be like cell cycle control or something like that, right? Well, again, you would be wrong, because the mechanism is another membrane-derived signaling molecule, not prostaglandins, but also involving the arachidonic acid system, which is the endocannabinoids, your brain's own marijuana. So the actual endocannabinoids are anandamide and 2-AG, and then they act through CB1 and CB2 receptors. They are ubiquitous throughout the brain. The CB1 receptor is considered one of the most abundant proteins in the brain. And they have lots of myriad different effects, which are very poorly understood during development. So one thing we did is ask, do males have more endocannabinoids than females in the amygdala? And they do, in fact, have a higher endocannabinoid tone. Remember, endocannabinoids are not neurotransmitters. They're just sort of continuously released and degraded, released and degraded, released and degraded. And males have more than females. You see here's the female here, and there's the male over there. This is a female treated with testosterone, and here it is blocked by flutamide. So this is an androgen-mediated sex difference that only lasts, again, for the first couple days of life. And then if we treat newborn animals with a CB1 or CB2 agonist versus antagonist, can we sex reverse this play behavior? The answer is yes. We give males, we reduce their endocannabinoid tone, and I'm just summarizing the data here. They play like girls, which that's not a bad thing. They're not sissies. They just don't play as much. And in the females, if we give them agonists for 4 days, test them 2 weeks later, they play like males. But again, it's nice pharmacology, but it's just pharmacology. The question is, what are the endocannabinoids doing? Well, come back again to our favorite cell type, the microglia, which in addition to responding and producing prostaglandins, also respond to and produce endocannabinoids. And here, when we took a closer look at the microglia, we found that they, in addition to making all these signaling molecules, they were also engaging in very high levels of phagocytosis. You can see these nice big balloons here. In fact, there was a new graduate student in the lab, Jonathan Van Rissen, who was looking at these microglia and said, what are those things? And the postdoc who'd done the first study said, I didn't know what they were, so I just ignored them. Oh my God, how could you ignore them? Those are clearly phagocytic cups. Here you can see is what I call our Baltimore lacrosse microglia. This is a common theme we see, this long arm and this big, huge cup. Here on the right we have our Mickey Mouse microglia. Here's our double D cup microglia. We had Beth Stevens visiting. We showed her these microglia. As you can imagine, we were like proud parents. Look at these microglia. And she was like, holy cow, those guys are not nibbling. Those guys are going to town. So is there a sex difference in this phagocytosis? Just again, looking just over the first 4 days of life, males are phagocytosing at a significantly higher rate than females. Look at these percentage here. We have about 25% of microglia at any given moment are engaged in phagocytosis. That's at any flashbulb given moment. This process lasts about 20 minutes to 2 hours, so there's an enormous amount of phagocytic activity going on. And if we treat females with endocannabinoid agonists or with steroids, we can completely sex reverse this phagocytic profile. So the big question is, though, what are they eating? Okay. Clearly they're eating other cells, but they're not eating just any other cells. They're very distinguishing diners. They are eating newborn cells. Right. About 60% of the phagocytic cups contain pCNA or other markers of newborn cells. And you can see here is this microglia. This is again what we see, this long arm. Here he is with his cup right around a newborn cell. And this microglia itself is also newborn, as you can see. Microglia aren't like neurons. They don't need 2 weeks to differentiate, and they're born hungry. Here's just another image of a newborn cell inside the cup of a ravenous microglia. And this is just repeating that point that they proliferate like mad. We also found about 40% of them are proliferating at this time, and then they are immediately gobbling up nearby cells as soon as they're born. Well, so they're eating newborn cells. Well, was that because those newborn cells were dying, or are they actually engulfing cells that would otherwise survive? And this is known as phagoptosis. It's a combination of apoptosis and phagocytosis. So cells will flip out what they call an eat me signal when they are distressed, and microglia can recognize that eat me signal, and it can come over and engulf a cell, and they just then kill it with an oxidative burst and ingest it. Or if the cell is left unmolested, it can put that eat me signal back away and say, I've changed my mind, actually life isn't so bad, I'd like to go on, and if they aren't phagocytosed, they will survive. So we wanted to know, were these cells that were otherwise going to live, or were they already destined to die? One way to ask that is simply to do a regression between the number of phagocytic microglia and the number of newborn cells using BRDU. This is from all of our experiments combined. As you can see, there's a very strong correlation between your percent phagocytic microglia and the number of newborn cells. But more specifically, we wanted to actually specifically block that phagocytosis. And we focused on the complement system for obvious reasons, and we used an antibody to block this complement system for the recognition of phagocytosis. And without going into great detail in the interest of time, let me just say that if we blocked phagocytosis selectively, nothing else about microglia activity, there were more newborn cells that survived in the males on the side that received the antibody versus the other side. Well, so a critical question is, what were those newborn cells destined to become? Were they just going to be other microglia? Is this just cannibalism, or were they going to be something important? Candidates are neurons, astrocytes, oligodendrocytes, microglia, or all of the above. Here we had to do fate mapping and wait at least 2 to 3 weeks to find out what they were going to be. And to our surprise, we were able to account for every single cell type that they were going to become. 80% of them were going to become astrocytes, only 13% neurons, and 7% microglia. So overwhelmingly what they are engulfing are future astrocytes. Right? And just to confirm that, we used here a precursor marker for astrocytes. You can see this microglia has engulfed a precursor astrocyte. It's glug, 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 glug down its gullet there as it is consuming this would have been an astrocyte. So when we think of all the things that can be modulated by sex as the brain develops, I think we have to add one more. So now you're going to say, well, wait a minute, where did the hormones go? She's talking about the immune system, and her talk is supposed to be about hormones. So that comes to my very last question is, how are these effects maintained from early development to adulthood? And for here, we turn to everybody's favorite form of cellular memory, epigenetics, 2 canonical forms, modification of histones, and methylation of cytosines proximal to guanines. And we focused on methylation of cytosines, which is carried on by DNA methyltransferases. There's DNMT1 and DMT3A and B, and they will recruit methyl groups to cytosines, which canonically can recruit methyl binding proteins, and then will just sterically inhibit transcription. That's not even epigenetics 101, that's kindergarten epigenetics, but I'm sure you all know it's far more complicated than that. But Bridget Nugent just asked a simple question, is there more DNMT activity in males versus females? This is again across development from birth up to 1 week of age. And here is the DNMT activity in a normal female, and here it is in a normal male, and here it is in a female that we've masculinized with steroid. So the steroid is inhibiting the DNMT activity. It lasts for the first couple days of life and then disappears after that. And all of the DNMT activity is shut down. So you can see there's a very narrow window here of a high dynamic period of DNA methylation. Well, does that actually translate into differences in methylation? We did whole genome-wide bisulfite sequencing of 9 animals, and these are the CpGs that are 100% methylated, and you can see here the females, all 3 females are above the males, and then here's a female that received masculinizing dose of estradiol for 2 days before. So in fact, the hormones are laying down the memory through the epigenetics that's going to endure into adulthood. This is all done in the preoptic area, by the way. Well, what are the genes that are being affected? Here we did an RNA-seq. We looked with and without DNMT inhibition. I'll tell you that overwhelmingly this was extremely disappointing. We found only one gene that made any sense to us, which was aromatase. And that was like rediscovering something that we've known for 50 years, only at $40,000 later. But then we did the IPA pathway analysis, and here we got an interesting surprise that what was different, in fact, in terms of pathway, is that the immune response genes are being epigenetically repressed in the females. So these are normal, healthy females, right? We haven't done any other manipulation other than inhibit their DNMT activity, and you can see that the genes that were changed are positive regulation of immune response, immune effector process, leukocyte activation, immune response, immune effector process, external stimulus, defense response, antigen processing, and presentation, right, in a normal, healthy animal. So So this would suggest that males have a higher inflamed state. This just confirms again that they have more immune cells, and those immune cells are more activated. The question is why? Why would there be increased immune activation in the males? If anything, steroid hormones are anti-inflammatory. They are not pro-inflammatory. So how can this be coming about? So we have a circular hypothesis that an increased immune activation in males decreases DNMT activity, which de-represses immune responses. But again, that's just it's chasing our tail, right? It's saying we still need some place that we initiate this difference in the males. And here, of course, we blame it on mom. And so what our working hypothesis right now is that the maternal immune system has to be repressed during pregnancy in order for the pregnancy to be viable, because you've got this foreign invader. But it is well known clinically and anecdotally that repression is not as strong against male fetuses as female fetuses because of the Y chromosome. And so we are postulating that the maternal immune system either directly invades the male fetus or attacks the male fetus with signaling molecules such as prostaglandins and cytokines, etc., that disinhibits the DNMT activity in the preoptic area, and we have data in the hippocampus. You demethylate or don't methylate the DNA, and that keeps the immune genes turned on, and then you have a positive feed-forward runaway process in which the male brain has more inflammation. Evolution works with the tools that it's given, and so evolution has co-opted some of these tools towards the normal masculinization process. So I can't emphasize enough that this is not a sick animal. These are normal, healthy males, but there's a big difference in their whole immune profile. So in conclusion, factors that are normally associated with inflammation or injury are elevated in multiple parts of the developing male brain, which I think makes them vulnerable, while epigenetic suppression provides protection to females. But that's developmental. Then in adulthood, there is a temperature inversion. Right? So this is a sort of a gender bias frequency chart. The size of each wedge of the pie is how large the gender difference is. So as I said at the beginning, the male biased ones are ADHD, autism, dyslexia, stuttering, Tourette's, early onset schizophrenia. As soon as we hit puberty, there's a complete conversion where females are likely to get anorexia, bulimia, multiple sclerosis, PTSD, anxiety, depression, and then a very unique late onset schizophrenia that comes in the 40s. It is 6-to-1 in women to men. So what happens, right? What is the shift at puberty? And that is the big question. And of course, in humans, environment and experience are likely dominant and impossible to control for. By puberty, women have had a lifetime of experiences already, and this is our big challenge: is how are we going to separate out the biology from the experience to understand their vulnerability, which appears later in life. That I'll just quickly thank my funding agencies and all of the excellent students and postdocs who have done the work, and you for your attention.
Great, thank you. We're a little ahead of schedule, in fact, so...
I thought with the interruptions I was late, so I was trying to rush,
So we had plenty of time for questions. Yes, Laurie. Do we have microphones?
Very behind.
Okay.
Ellen, yep. Is Ray available?
Why don't I take one? I'll distribute to the front. Yeah.
Hi, very, very interesting talk. I was wondering if you could talk a little bit about some of the specific chemicals you talked about in windows of plasticity. So, taking things that modify arachidonic acid or other things later on in life, can that have effects on brain and behavior?
Yeah, so I think what you're asking is about pregnant women taking COX inhibitors. Would that be?
That or any other thing.
Yeah, so certainly the animal data, would suggest that we've treated pregnant animals with aspirin in their drinking water, or we've treated them with indomethacin, which is the go-to drug to block premature labor in humans. We treated them with PGE2 directly, which is used for patent ductus arteriosus and stuff. So we know in our animal models we can have these permanent effects. The question is, can we be having them in humans as well? And there it gets enormously challenging because the predicted impact is decades later, right? And also in terms of the preoptic area and the male sex behavior, those effects are very, very specific to male sex behavior. And we're not trying to model sex behavior in humans, right? We're just trying to understand unique ways that the brain develops. Nonetheless, I started to look into it, and I worked with the Kinsey Institute and various places to how would we, could we use the priesthood? Could we look at prisoners? How could we get to it? And it turns out that all the tools for studying male sex behavior in humans involve penile tumescence because they're all based on Viagra. And there's absolutely no way to measure male sexual motivation that is considered a useful tool. So we've really been confounded in our ability to actually make that connection. In terms of the endocannabinoids, the big question is what about marijuana use during pregnancy, right? There have been relatively few studies on outcomes from marijuana use during pregnancy. They do find some social deficits in some studies, but not in others, but they're really very few. The problem is that most of those cohorts are from when marijuana was a much different drug than it is today. Right, in terms of potency, right? You couldn't tell oregano from marijuana kind of thing, and now it's like the THC and cannabidiol contents are completely different. So again, we really don't know yet. We have to wait and see. But part of the goal is if we can, in our animal models, at least highlight, because there's a fear that there's a perception that marijuana is perfectly safe to use during pregnancy. It is the most commonly illicitly used drug during pregnancy. Alcohol is the most commonly used substance, but marijuana is the most illicit used drug. And boy, we're just really in a black box of information.
Yeah, the question, yeah. Can we get the microphone?
Oh, no, wonderful talk. Could you comment very briefly on the effects of viral infections during pregnancy on the system and various neurotransmitters and
Yeah.
Do you want to repeat the question?
Oh yes, indeed. The
Thank
question
you.
was the impact of viral infections during pregnancy on the various endpoints that I looked at. It's an excellent question. I don't have a good answer for you because, again, we have not done maternal immune activation yet. It's one of those things that's on our agenda to do, but we haven't. What we have done, I guess maybe this is, but I should say what we have done, and we're just submitting for publication now, is we've made rats allergic to egg whites. And then we've gotten them pregnant, and then we've caused a massive allergic reaction while they're pregnant at the time that sex differentiation is happening. And what we've seen is that the female offspring are in fact masculinized. Again, it's normal. They're not abnormal. They just show normal male behavior. But the males are actually dismasculinized, almost like it was just like too much. We showed that it increased the mast cells in the offspring of both the males and the female fetuses. It increased their degranulation, etc. So something even as benign as an allergic reaction can tap into these systems. The question is if a viral infection, which has its own unique imprint, might, is it hitting the microglia and having something different type of effect? Yeah, we just don't know that yet.
I think we're going to have the next video.
A quick question to neurogenesis and gliagenesis in male rats that we found very early in development to be increased. Does this continue to adult neurogenesis? Does this keep up? Do you see more inflammation and more dendritic branching or such thing in the hippocampal area?
In the hippocampal area?
Yes.
I didn't. Okay.
So the hippocampus is the area in which you contain into adulthood neurogenesis.
Yeah, I understand what you're saying. Yeah, there are other individuals who've done a lot of work on adult neurogenesis, and it is different in males and females, but very complexly controlled by the hormonal milieu, which of course is varying in females and is different. One of the problems with doing like a direct sex difference in adulthood is you're really comparing apples and oranges because of the difference in the hormonal milieu. And the amygdala, I should have emphasized the point that what we thought was a sex difference in neurogenesis actually turns out to be a sex difference in phagoptosis. They are probably proliferating at exactly the same rate, but they're just dying in the male. Not dying, they're being eaten in the male by those microglia, eaten and killed. We also see that we have a sex difference in hippocampal neurogenesis that I didn't talk about, and it only lasts the first 6 days of life, and then it goes away. Males make about twice as many new neurons as females, but then it's gone. Everything we see is just this narrow first 5 days of life. It's a very short window, but it's a real important one for a lot of sculpting of the brain development.
In the amygdala?
The source of the endocannabinoids? Yeah, we thought it might be the microglia themselves, but we depleted them, and it had no impact on the anandamide and 2-AG content. So it's not the microglia. And we think, so the question is it those newborn cells? Are they actually committing suicide? Are they calling the microglia in to gobble them up? Or we've also been entertaining the idea that the newborn cells are actually calling out their neighbors. Like, did you get to it? So they're clearly very tightly regulating the number of astrocyte precursors. And are they saying, okay, there's enough of us now, let's turn down our endocannabinoids and stop this slaughter? Or is they also saying, like, get that guy over there, and they're sending out endocannabinoids? So we're trying to look at various like competition hypotheses, group selection hypotheses. But it's critical for us to know who's making the endocannabinoids. And the problem is the neurons and the astrocytes are probably almost definitely making it. Everybody has both the CB1 and CB2 receptor as well. We've done all that. And developmentally, the endocannabinoid system is much, much more poorly understood than in the adult. And they're like both CB2 and CB1 are hugely relevant, whereas in the adult you mostly have only CB1. So it's kind of anything goes right now. It's exactly the kind of thing we're trying to figure out.
Question from
Thanks, that was an awesome talk, amazing. I was just wondering, my talk is like silks, I think. I'm interested in brain region differences, and in your prostaglandin study, did you inject into the preoptic area, and did you ever try injecting into other areas, and did you see any similar?
So we did inject into the preoptic area, but in truth in advertising and these really small animals, there's no way it was limited to the preoptic area because it's such a small brain region, and so we probably flooded the whole brain. But I'm glad you asked the question about region specific, because we did look at see if prostaglandins was mediating the effect in the amygdala. No. We looked in the hippocampus for cell genesis. No. We looked in the arcuate for a different study. No. The VMN. No. So it's very, very tight to that region. Same thing with the microglia phagocytosis in the amygdala. That's very specific to the posterior dorsal medial amygdala. If we even looked at other parts of the amygdala, it's not there. So there's some kind of rules that are setting up in very tightly constrained brain regions. And I used to give a talk called The Building Blocks to the Male Brain, because that's the way I would view the brain, is every little region is completely separate and independent, all driven by steroid hormones and maybe the immune system, but something limits the effects to that region. So there's no sort of unified field theory for sex differentiation in the brain, which is disappointing but also exciting. What it does, though, is it makes it so there's no such thing as a male brain and a female brain, because you have so much heterogeneity. You have sources of genetic variation, environmental insult, local factors and everything. So you could have an amygdala that's highly masculinized in one male and not so much in another, and you could have a preoptic area in a female that's a little bit masculinized and maybe a little. So you get this mosaicism, and I think that actually, in retrospect, nature designed it that way because it gives much more phenotypic variability in terms of maleness and femaleness, so that it's not just a unitary, even though the brain is in a body that is male or female, the brain itself is a mosaic mix of male and femaleness and sameness.
Maybe two more questions. Ursula and then
That was fantastic.
Thank you.
I really enjoyed that. And I have a million questions, but I'll limit myself to one. So, can you comment on, you talked about the role of gonadal steroids. Can you comment on the role of neurosteroids and whether there are sex differences in neurosteroids and the relative importances?
Yeah, it's a good question because particularly like you think.
Sorry, can
Oh, I'm sorry.
Everybody at the back, I'm not sure that the mic is activating. Can you all hear the questions at the back? No. Okay. Maybe you could repeat.
So quickly, the question was neurosteroids and whether or not they might be playing any role. And let's define neurosteroids as like the allopregnanolone, GABA receptor modifiers and things of that sort. Yeah, that's a really good question. I would admit we have not looked at that carefully, although we've looked a lot at the target of the neurosteroids, the GABA receptor, particularly because GABA is depolarizing and excitatory at this time, and we have found estradiol modulation of the excitation effects of GABA, but we've never been able to tie that to any functional readout. The neurosteroids are so hard to measure. They're such a bear, right? And so that's also why we haven't really gone there. But it's a good question. It could easily be an important modifier.
In presentation as well. I was wondering if you have any idea whether this phagocytosis also happens in the preoptic area, because we know that different neural populations happen in the female, like Kiswan neurons, for example, in ovulation, are not present in the male. So I was wondering if maybe prostaglandins have a role in the get appearance of these neurons?
Yeah, thank you. Excellent question. So the question is whether or not there might be phagocytosis in the preoptic area as well. And as I said, when the original postdoc did the study, she just completely ignored them. But if you even looked at those images that I showed there, you could actually see there was a lot of phagocytosis going on in the preoptic area. We are actually doing those studies right now. If my graduate student would stop having babies and going on maternity leave, no, really, it's fine. It's okay.
We're not allowed to say that.
I know. She is finding this amazing data around the sexually dimorphic nucleus of the preoptic area, which looks like it's being sculpted by phagocytosis. So we have not thought to look in the AVPV, and that's an excellent suggestion, and we should perhaps be doing that. And it's probably in a similar section. So that story is interesting because of the mast cells in that region are also regulating the phagocytosis, but in a completely different way. They seem to be inhibiting the phagocytosis. So it's stay tuned. We hope that's going to turn into a very interesting story.
Great. Well, thank you very much. That was wonderful.